The search for extraterrestrial genomes is a project to search for life beyond Earth. In particular, the most timely place that we might look for life might be Mars. And this project got started because of a series of studies that were carried out in the late 90s. And these studies revealed that there had been extensive transfer of rock between Earth and Mars due to large meteorite impacts. And as a result, if life was around when these impacts had occurred, which is about 3.5 to 4 billion years ago, during a period called the Late Heavy Bombardment, that life could have been transferred between the planets. So, there's many assumptions that are built into that. Life would have to be around then. It would have to survive a large meteorite impact that would eject material, say, from Mars to Earth or Earth to Mars. It would have to survive that ejection. It would have to survive the transfer in interplanetary space radiation during that time, and then it would have to survive the re-entry into through the other planet's atmosphere, and then it would have to survive and propagate in this new environment. So that's a lot of requirements, but over the time since these studies were carried out, many of these requirements have been tested both theoretically and empirically. For example, it's been shown that microbes can survive the kinds of shock pressures that would be encountered during a large enough impact event to eject material into space. And it's also been demonstrated that some of the material is ejected in a way that's low temperature, so it wouldn't be sterilized. Furthermore, it's been shown that some of the material, a small fraction of the material, can be carried between Earth and Mars over very rapid timescales. And here I'm referring to Mars to Earth, but it could happen the other direction as well, where you would get transport even within a few years for a small fraction of that material. So that could help with surviving space radiation. Of course, we also know that many organisms, for example, Deinococcus radiodurans, can tolerate extreme levels of radiation. It can live in nuclear reactors, for example. And then in terms of surviving arrival at a new planet, the speed at which these rocks would be traveling is such that they would go through the atmosphere very quickly, maybe in under a few seconds might be typical. And so in that context, they would be heated an extraordinary amount, but only on the surface. So they will be burned to a crisp on the surface, but inside they might still be at the temperature they were at in deep space, which could be just a few Kelvin. So in that context, people have actually found recent meteorite falls where the surface of the meteorite was covered in frost. And so this indicates that the center of that meteorite was still quite cold after it passed through the atmosphere. So in this way, any microbes that, say, were hiding within fractures of this rock wouldn't be sterilized. And the timing of the Late Heavy Bombardment, 3.5 or 4 billion years ago, was a time when both Earth and Mars were thought to be habitable. Of course, impact events might have made the planets less habitable, but they also create niche environments where life as we know it could have taken root if it was arriving from beyond Earth, for example, or life arriving from Earth and ending up on Mars. So the big idea is that life on Mars, if it in fact exists, could be related to life on Earth. And that's why we are proposing to go and search for life on Mars that's very similar to life on Earth. Basically, we would like to look for RNA and DNA. And so we're building a small sequencer to send to Mars and look for life. And I should add that this project is a collaboration between Gary Ruvkin here at Mass General, and he's the one who started the project, submitted the original proposal to NASA, and also Maria Zuber at MIT. And Maria is the one who recruited me to the project back in 2005 when the money from NASA was arriving and we were ready to set about our work. So we've been working since then to figure out, what protocols would we use, how would we construct this instrument, what are the right technologies to use. I've given you a little background about why we would search for RNA or DNA. One of the, an absolute gold mine, very unlikely, but the absolute gold mine would be to go and look for life on Mars and to find something that is very ancient. It's been very dry and cold for about 3.5 billion years. And so in that context, life there, if in fact it is there, would maybe not have evolved as much away from its ancestor as on Earth. So we might be able to sample a very ancient environment by going and looking for life on Mars. And it's thought that before the current DNA world, that there was an RNA world where RNA served the role of both information and function, just as some of the core RNA machines like the ribosome do in all known life today. And that's a vestige of this RNA world. So one dream would be to find the RNA world on Mars, although that's probably very unlikely. What we'd like to do is build this instrument, send it to Mars. We've been studying the different technologies we could use to do that. And so we originally were going to design an instrument based on PCR, or polymerase chain reaction, where we can take a piece of DNA and target known regions in that DNA with primers that are basically pieces of DNA complementary to, say, conserved regions in a genome, and then amplify the unknown DNA between them. And this is the standard way in which people prospect for life here on Earth in every environment, from the most benign to the most extreme. It would be called barcoding, or just PCR-based barcoding. And in this way, we can characterize who's in a given environment. It gives us a lot of information, but sometimes you could envision that life may not have this same conserved regions that we target with these with PCR, with these primers. And so we went and looked at all the different organisms across the tree of life and identified, what are the most conserved sequences. And as it turns out, they are the sequences, or in the same genes anyway, that people have been targeting for some decades now. They're regions within the ribosome. But there are also now counterexamples where microbes might have a base that's different in these universal regions. And some of these microbes are the most interesting. For example, the Banfield lab has identified nanoarchaea in the Richmond mine that are extremely small cells, and they have a mismatch in this canonical region. So these archaea have a mismatch in the canonical universal region of their ribosomes, and that means that a PCR-based approach might not find these organisms. So with that in mind, we set about making our approach to life detection more general through metagenomic sequencing, which in this context means sequencing any RNA or DNA. And in parallel, the technology for sequencing has been rapidly evolving since, certainly since 2005 when we started this project. That led us to look for technologies for massively parallel sequencing. So in that context, we can go to Mars, look for any RNA or DNA, start with a soil, ice, or brine sample, isolate nucleic acids from that, and then we could do a PCR, but we can also just sequence any nucleic acids. And so one of the technologies we identified that would be eminently compatible with the Mars application is this chip here, which has been commercialized by IonTorrent. And the basic principle behind this is to use the natural chemistry when a nucleotide is incorporated into a growing double-stranded DNA molecule, a hydrogen ion is released. And so if you can imagine, this chip has 6 million wells, but if you can imagine one well on the chip, what we do is we take beads and we amplify at single molecule. Imagine a bead here, and we try to do a reaction, a PCR, where we have a bead and one DNA molecule in its own little reactor vessel, an emulsion droplet. And that's the ideal, and it doesn't always work out that way, but we try to amplify. So now we would have 100,000 or a million copies of the same DNA molecule on the speed. Then we take those beads and we load them into that chip, and then we make the second strand of the DNA. And this is all the standard ion torrent sequencing approach. And when we do that, now when you get a base incorporation, you flow the different nucleotides by the A's, the C's, the G's, and T's. And if you get a base incorporation, now instead of one hydrogen ion, you'll get 100,000 or a million. And that produces a change in pH that can be detected by an ISFET, an ion-selective transistor at the base of each well. And then you can turn a DNA sequence into essentially an electronic readout. And so that's what's so fantastic about these chips for the Mars application is we have an electronic readout of a DNA sequence. It doesn't involve optics. It's inherently small. It's also robust, and we've specifically taken these chips and exposed them to analogs of space radiation and shown that at the doses we would expect for Mars that they would, there's essentially no impact that we can measure. We actually can measure manufacturing variability, but it's not even significant in terms of DNA sequencing or error rates. So this is a great technology for us, and of course we'll continue evaluating as the market develops. But we've demonstrated that we think we can do this on Mars. The sample preparation area is one of the hardest areas, and that's something we're actively working on. NASA has funded this project so far since 2005. We hope that we'll get to the point where we can get on a mission. Maybe the earliest time we could be on Mars would be 2022. Our intention is that this would go on a robotic mission. It's certainly more simple and less expensive to envision a robotic mission to Mars in this time frame. This instrument could be used by humans as well, and in fact, we think that this same instrument would be a valuable device to take on a human mission, because you could use it for diagnosis or monitoring your environment, or also for potential synthetic biology applications. You know, this instrument could be used by humans, but we envision it on a robotic mission. Human exploration may occur in the 2030s. NASA's current plan here in 2014 is for humans to go to Mars in the 2030s. But that timing of a human mission to Mars has been kind of a moving target. And the reason that we're targeting 2022 is that it really takes a long time from when instruments are ready until they're on Mars. There's a really intense development cycle to get all these instruments integrated and validated, tested, and finally integrated into a launch vehicle and launched to Mars. All these things take a long time. In fact, even the preliminary development takes a long time. I think the average instrument takes 10 or 12 years before it's selected for a mission. So we're at about 8 years right now. So we're doing pretty well. We've got a few more years, but we think we'll be ready pretty soon.